With climate change, temperatures throughout the world are becoming more extreme and unpredictable, adversely impacting entire ecosystems. At a smaller scale, farming is particularly affected by this drastic weather. For instance, in the Swiss region of Valais, we observed the worsening of spring freezing spells in the past few years, causing devastating losses in crops. According to our expert, Dr. Danilo Kristen, the last 2 extreme freezing spells within the past 5 years caused devastating losses in apricot crops specifically. A staggering 85% of apricots were lost this year. This dramatic outcome is an important cultural loss for our region, as it endangers the celebrated apricot du Valais, traditionally consumed each summer. It also threatens the livelihood of all those involved in the cultivation and processing of apricots. We are the University of Lausanne's 2021 iGEM team, and we dedicated our project, ApriFreeze, to the protection of apricot trees during freezing spells. Addressing this problem is urgent and of great importance locally. However, the use of the systems we designed to combat this problem would not be exclusive to apricots. Our technology could be diversified to all types of crops and could be used all over the world, as losses due to frost are a global occurrence. We first studied the cause of these devastating losses. Of course, low temperatures play a huge part, but the presence of a microbe on our apricot trees exacerbates the damage due to frost. Water does not immediately freeze at below freezing point temperatures. It requires a catalyst, called an ice nucleus, on which a crystal starts to grow. Plant tissues themselves are relatively bad ice nuclei. When frost-sensitive plants are grown in sterile conditions, most of them can resist temperatures as low as 5 to 10 degrees below Celsius without frost damage. However, certain bacteria, such as Pseudomonas syringae, synthesize proteins that act as good ice nuclei and therefore promote ice crystal formation. These microbes can therefore increase frost damage on plants, especially barely below freezing point. For instance, Pseudomonas syringae produces a membrane protein called INAZ to induce ice nucleation. To combat the frost damage on apricot trees, we created a system to address the problem on 2 fronts. The first was to protect the apricot trees from damage caused by ice crystals formed at low temperatures generally. To do so, we envisioned coating the plant tissues in a solution of antifreeze proteins. These proteins bind to the crystals and inhibit their growth, diminishing the harm done to the cell. The second front on which we wanted to attack this problem was to reduce the damage caused by Pseudomonas syringae, the pathogen present on apricot plants that exacerbates damage during freezing spells. We had 2 ideas to combat this pathogen. Our first was to simply eliminate it by using tailocins. Tailocins are protein complexes expressed by bacteria that resemble phage tails and allow the specific killing of a target strain. Our second idea to diminish the impact of Pseudomonas syringae was to inactivate the gene coding for the ice nucleation protein. By genetically modifying the bacteria in this way, these pathogens would no longer be able to produce the harmful protein that causes the worsening of frost damage. Now we can have a closer look into the design of the strategy we plan to use to inactivate the INAZ gene. We plan to inactivate this gene using CRISPR-Cas9. We designed 3 different single-guide RNAs targeting the INAZ gene that, together with Cas9, should induce a double-stranded break in the genomic locus. The broken DNA would then be repaired via homologous recombination, a mechanism naturally found in Pseudomonas species. For this, we designed our CRISPR-Cas9 phage mid to bear homologous region directly flanking the INAZ gene to be deleted. These homologous regions belong to a structure called donor sequence. Although we knew what method we wished to use to edit the genome of Pseudomonas syringae, we were unsure of the best way to implement it. We therefore turned to experts who directed us towards the use of a phage mid as a means to deliver our construct into Pseudomonas syringae. We envisioned transforming E. coli with 2 DNA structures, our engineered phage mid and a helper phage. The helper phage is a plasmid-like structure carrying the genes necessary for the construction of new viruses. The phage mid also resembles a plasmid, but only carries a packaging signal and the constructs we aim to deliver to Pseudomonas. Once inside the host, the helper phage sequences are expressed and new viral particles are formed. Alongside, the phagemid is replicated. Phagemids are then recognized and packaged into the newly formed viral capsids, starting with a pack site. The virions generated only carry the phagemid and not the helper phage sequence. Because of this, the constructed virions cannot be reproduced by the infected Pseudomonas. This method is a safer approach as it allows us to avoid outbreaks of this bacteriophage. However, these viruses could modify the INAZ gene in Pseudomonas syringae, effectively creating a GMO. The use of GMO in agriculture is currently forbidden in Switzerland, making the phage part of our project impossible to implement here at the moment. This isn't the case in other countries, such as the United States or countries in the European Union, which is why we thought it was still a promising strategy to explore. For more information on this concept and the work we undertook on this ambitious project, check out the wiki page. As an alternative to inactivating the gene encoding for the INAZ protein by using GMOs, we thought of using tailocins to simply eliminate the pathogen altogether. Tailocins are protein complexes that have a similar structure to phage tails. The expression of these tailocins allows bacteria to specifically kill other bacterial competitors. For this, the killer bacteria first have to express the tailocin genes and produce them. Producing bacteria then lyse and release the tailocins in their environment. These tailocins can then bind to the cell wall of the target bacterial strains and puncture them, effectively killing the cells. We found tailocins to have great potential for our project. They have a narrow killing range, which would allow us to target our plant pathogens without harming any other bacteria present on the plant. Another advantage of tailocins is that, contrary to phages, they are not replicative. They are simple protein complexes that are also conveniently not considered GMOs. In the context of our human practices research, we were warned that the use of GMOs in agriculture is banned in Switzerland. Tailocins therefore represent a great means to kill Pseudomonas syringae, as it was important to us that we designed a solution that could realistically be implemented locally in accordance with legislation. As a model for the Pseudomonas pathogen found on apricot trees, we used Pseudomonas syringae B301D, a strain closely related. We produced tailocins using the killer strain listed here, as it was previously shown that this bacteria expresses tailocins that specifically kill our target strain. To induce tailocin expression in our killer strain, we exposed it to mitomycin C, an antibiotic which causes double-stranded breaks in DNA. These breaks trigger a stress response, resulting in tailocin production. The tailocins are then released with the lysis of the cell and are able to attack the target strain. This slide shows the result of adding mitomycin to a killer strain culture. In the control, which we did not induce, you can see that the solution is rich in bacteria. On the other hand, adding a concentration of 0.5 or 3 micrograms per milliliter of mitomycin causes cell lysis, which indirectly indicates tailocin production. This demonstrates that even low concentrations of mitomycin can induce the expression of tailocins. Having successfully produced tailocins from our killer strain, we then purified them from the bacterial lysate and tested their efficacy in killing our target bacteria. To do so, we placed drops of purified tailocins with decreasing concentrations on a soft agar overlay of our target strain. We observed significant lysis plaques after induction, which allowed us to conclude that our tailocins were functional. In addition, we also observed our purified solutions by electron microscopy to confirm that the killing agent present in our solution was in fact our tailocins. This allowed us to characterize our tailocins by obtaining their dimensions. Although we were able to show that mitomycin C was an efficient way to produce tailocins from Pseudomonas syringae, it was both expensive and toxic, which limits the possibility to produce tailocins at a large scale. To make the implementation of our product cheaper and safer, we wish to produce our tailocins from E. coli instead. To do so, we would clone the tailocin cluster in E. coli under the control of an inducible promoter. This would allow heterologous expression of tailocins in a regulated way so that we would avoid the use of mitomycin altogether. We have started the work to reach this goal and have laid the foundation for future iGEMers to complete this. For more details about this, you can check our wiki. We were very excited to have successfully produced tailocins that could be added to our spray-on frost protection solution. But we wanted to go further, and so, in parallel to these efforts, we worked extensively towards obtaining antifreeze proteins that could be coupled to the use of tailocins. This way, tailocins could kill Pseudomonas syringae, and the antifreeze proteins would prevent the negative effect of any surviving cells or of the low temperatures in general. Antifreeze proteins have two important properties: thermal hysteresis and ice recrystallization inhibition. Thermal hysteresis describes the difference between the freezing and melting temperatures of a solution containing antifreeze proteins. Below the freezing point, ice crystals grow, and above the melting point, ice crystals shrink. Between these two temperatures, ice crystals neither grow or shrink. Ice recrystallization inhibition is the protein's ability to prevent the growth of larger ice crystals that are the most damaging to plant tissues. The combination of these activities, which vary depending on the protein structure, prevents the freezing of body fluids and cell damage in organisms that live in environments with extremely cold temperatures. For this project, we chose to work with 3 antifreeze proteins with varying properties. This allowed us to compare the properties of proteins in order to find the optimal combination for our product. The main approach we used to produce these proteins was to clone them in E. coli BL21, using 2 different vectors, PET17B and pCold1, and then to induce expression with IPTG. Finally, we extracted the purified proteins using magnetic beads or a histag affinity column and gel filtration. PET17B contains a T7 promoter, which allows regulated high-level expression of recombinant proteins. On the other hand, pCold1 includes a cold shock protein A promoter, which allows expression of proteins that are more difficult and cannot be expressed with the T7 system. Therefore, we compared expression of antifreeze proteins using each vector with the aim to optimize expression and synthesis of antifreeze proteins. We successfully cloned various AFP genes into each plasmid, as shown in the figure, and confirmed by Sanger sequencing. The next step was to express our AFPs from each construct. We first grew liquid cultures at 37 degrees, then induced AFP expression and compared how the promoters of our vectors influence expression efficiency. The expression was successful for FFIBP and RAAFP, and the pCold vector worked much better. The bands on the gel appeared to be thicker for this plasmid, so we concluded that the pCold plasmid was a better expression system for cold-stable proteins like AFPs. We then moved on to purify our antifreeze proteins by using the histag affinity column and gel filtration. Our gels showed that the purification was successful. We were thrilled about these results and then wanted to test if the antifreeze proteins could actually prevent freezing. From our human practices interviews, we learned that it would be legal and feasible for us to implement our antifreeze protein solution and tailocin solution in Switzerland. We therefore decided to focus our proof-of-concept efforts on testing these solutions, as our main goal was to solve this problem locally. To compare how the freezing temperatures of our protein solutions vary compared to a simple buffer, we built a low-budget self-made device called Frozone. This machine can change the temperature precisely and allow us to track the freezing and melting points of drops of our various solutions. The core of our device is composed of a thermoelectric cooler. By using an electrical current to create a heat difference between each face of the thermoelectric module, we were able to lower the temperature of a copper plate to about -20 degrees Celsius and freeze drops of samples. We also added a vacuum chamber to the machine to minimize ambient humidity from crystallizing on our samples. The whole process is controlled by a custom Python software, Edna. You can learn more about the details of this device on our wiki. Samples are placed on the cooling copper plates of our device, and freezing and melting temperatures are determined by microscope video analysis. We used Frozone to confirm that the presence of Pseudomonas syringae in a sample greatly diminishes the time it takes for the freezing to occur at -5 degrees Celsius. We were able to show that the pathogen does have an ice-nucleation activity. We also used our device to test the efficacy of tailocins in killing our target strain of Pseudomonas syringae. We added tailocins to a solution of this bacteria and showed that this greatly increased the time it took for a drop of solution containing the pathogen to freeze compared to the time it took for a control containing only the pathogen to freeze. To conclude, we were able to demonstrate that tailocins successfully counteract Pseudomonas syringae's ice-nucleation properties. To determine how often tailocins should be applied to the plant to kill the pathogen strains, we decided to simulate the effects our tailocins would have on a population of our target bacteria. To do so, we made a model based on the Lotka-Volterra model. We were also able to measure the thermal hysteresis of our antifreeze proteins with this device. To do so, we simply measured the freezing and melting temperatures of drops of antifreeze protein solution at various concentrations. This allowed us to determine our purified protein's activities and to confirm that they are functional. To determine the optimal concentration that is needed for a protein to function on plants, we used a kinetic pinning model to simulate the thermal hysteresis at increasing concentrations. This also allowed us to compare our measurements to that of literature. We also wanted to test if these proteins could truly protect plant tissues from damage. To do so, we incubated Arabidopsis thaliana plants at -5 degrees Celsius overnight in an antifreeze protein solution or using a control treatment. We then stained the leaves using trypan blue, a dye that enters damaged cells. Using our image processing software, Vision, we calculated the fraction of damaged surface on the leaf. We were able to demonstrate that plants treated with our antifreeze proteins had significantly less damage than the control group, showing that these proteins protect plants from damage caused by ice crystals. We discussed the implementation of our project thoroughly with experts and concluded that the use of our tailocins and antifreeze protein solution was theoretically possible in Switzerland. They would be particularly useful on the trees grown on mountainsides, as current solutions to combat frost damage in these areas are largely ineffective during severe freezes. Our product could be sprayed on the plants using a tractor. This solution would be financially feasible as apricots are a huge source of income, so farmers would be able to invest in their protection. In the context of the education part of our project, we first decided to produce a 4-episode podcast on GMOs. We did so in collaboration with the EPFL team. In this podcast, we discussed the topic of GMOs with various experts and governmental representatives. We kept a purely factual tone to our podcast, as the aim was not to influence our listeners, but to inform them. We also held a weekend-long event for high school students to allow them to discover synthetic biology. This program was focused on teaching our students to understand and perform the lab work we spent all summer mastering. This concludes the highlight of our iGEM project. Thank you so much for watching. Lastly, we would like to thank our sponsors and everyone who supported us throughout this project.